Okay, so let's then move on to the reading brain. So what do we know about the neuroscience of reading? Just like in the auditory domain, I just want to take a quick look at the relevant pathway first, so we have some understanding of how the signal gets, from your eyes to the cortex. So just like in the auditory domain, the visual pathway goes through the thalamus. So you go from the sensory organ through the thalamus to the relevant sensory cortex, which is for the case of vision at the back of your head. So occipital cortex is all the way sort of on the opposite side of the head from your eyes. The kind of fundamental property of the visual pathway is that it exhibits this crossover. So if you're fixating in the middle, then a visual stimulus that's is in the left visual field, so to the left of the middle area, which is what we call the foveal area, that stimulus is going to be projected to your right occipital cortex. And similarly, if the stimulus is to the right of the foveal field, so in the right visual field, that stimulus is going to be projected to your left visual cortex. And the material that is in the middle is foveally presented. The textbook understanding of that is that that stimulus is going to be bilaterally projected to both visual cortices. So the visual pathway doesn't exhibit quite a similar redundancy as the auditory pathway. So remember, the auditory signal from each ear is projected to both hemispheres. In the visual system, signal from one visual field is only projected to one hemisphere. The visual pathway is also slower than the auditory pathway. Precise timing is controversial, but estimates vary between 50 and 80 milliseconds for visual information to reach visual cortex. Okay, so we want to talk about the cognitive neuroscience of reading. And so the cognitive neuroscience of reading should be considered as one part of the cognitive neuroscience of higher level vision in general. So it's kind of one branch of the vision sciences broadly construed. And the question that has attracted a lot of attention over the years is whether letters and printed words are special kinds of visual objects for our brains. So just like in the speech domain, we're asking the question, is speech a special kind of sound for the brain? Similarly, in the visual domain, many researchers have investigated the parallel question intensely. So do our brains do something special with letters and printed words? And interestingly, much of this literature has happened in the context of another literature that really isn't about language. The cognitive neuroscience of reading, in some sense, stems off from the cognitive neuroscience of face perception. So why is that? I'll try to make it clear. Specifically, there has been a finding originally from Nancy Canwisher in 1997, where it was discovered that the right fusiform gyrus, now we're at the bottom of the temporal lobe, there's a response there that shows selectivity for faces as opposed to other types of visual categories like hands, houses, animals, and so forth. Pretty much any control condition that you can think of has been tested by now. So here we're looking at the bottom of the brain. So if you imagine the bottom of my brain like this, and here we see the fusiform gyrus. There's actually a lot of cortex, and usually within the fusiform gyrus, there's a specific spot for each individual that shows this face-specific response. So it's like a specific area of the brain just gets much more excited about faces than other categories. So that was a very interesting category selectivity that was observed. So here you can see some examples of those stimuli. So So more signal, so that's the kind of spot that you would find. So more signal for faces than for objects. And all the kind of right visual controls have been tried. So you would scramble the faces in order to keep all the low-level properties the same, and then just sort of vary the holistic perception. And there's also another patch of cortex that shows a bigger signal for objects as compared to faces. So this area is an area that we now call the fusiform face area, FFA. Anyone who works in the cognitive neurosciences knows this acronym. Very influential, famous work. Okay, so what does this have to do with reading? Well, so faces have been around forever. So it is evolutionarily plausible that we actually would have evolved to have this kind of face area in our brains. There's been enough evolutionary time for such an area to develop. However, we've only been reading a few thousand years. So first writing system was the Sumerian script. Sumerian scripts, roughly 3,500 BC. It's roughly contemporary to the sailboat and a wheel, if you're curious. But in evolutionary terms, writing is a very recent innovation. So it just doesn't seem likely that there's been enough time for some specific region of the brain to evolve to read. And so that's kind of like the starting point for people's curiosity of like testing whether we might have such a region. So it would be very surprising because it really doesn't seem like we've had enough time to develop that kind of a region. But lots of studies have looked into it, and turns out that it seems that we do have a region like that. So the results have actually been quite surprising. And very interestingly, the region that seems to like visual words more than other categories is also in the fusiform gyrus, but just in the other hemisphere. So actually in the language hemisphere, the left hemisphere. And so here we have a visualization of that area. We call it the visual word form area. VWFA, that does not roll off the tongue nearly as well as the FFA. So usually people say visual word form area. And here's a figure from a review article by McCandless, Cohen, and Dehan. Much of this research is from this research group showing the localization of that region. This is neuroimaging data, so not timing data, just spatial data. And so here's the broad characteristics of this region. So it's activated by words and pseudo words. So words like actual words like table, but then also pseudo words like blicket or mide. So strings that could be words in your language, so look like existing words, but just doesn't map onto a meaning. And so that activation is more than other similar control stimuli. For example, just like a consonant string that does not have the kind of bigram transitions that you would have in an existing word. And it seems to be specific to the visual modality, though this statement is controversial. The data on this generalization are a little bit messy. It's possible that it's actually adjacent to another area that does show a more multimodal character, so responds more also to auditory words. But generally, we think that there is at least some patch of cortex that seems to be specific to the visual modality. And just like we would expect from a region that represents letters and word forms, it is insensitive to variance in the sort of visual specifics of the stimulus, such as case, for example. So we have a fair amount of evidence that, for example, repetition suppression in this area does not depend at all on case. the aphasia literature is nicely consistent with the hemodynamic literature. So we know that if your visual word form area is damaged, that often leads to so-called pure word alexia. It's a type of aphasia in which you have a deficit in reading while you can still name objects just fine. So if you're given a picture of a cat, you can say cat, but if you're given the orthographic form, you won't be able to read it very well. So keep the triangle model in mind that we talked about last time. So these patients actually seem to engage in the type of reading that we know fluent readers don't do. So the type of letter by letter serial reading that has been ruled out as a kind of a good mechanism to explain fluent reading behavior. These patients seem to engage in letter by letter reading, and so their parallel letter perception seems to be impaired. So their reading is consequently very slow and laborious. Okay, so that's a quick summary of what we've learned from hemodynamic methods and from aphasiology. But now to really understand the visual word form area, we need to get some timing information about its activation. Otherwise, we don't really understand what's going on. So our suspicion would be that it's relatively early activation. You have to kind of parse a visual word form before you can access meaning, for example. So our expectation would be that the visual word form area should be active relatively quickly after the onset of, say, a visual word. But we need some temporal resolution in order to actually test that, and so electrophysiological techniques offer that to us. So, for example, with MEG, we can measure activity elicited by visual words millisecond by millisecond. So here we have our participant, and the MEG sensors are in the doer collecting the strength of the magnetic field millisecond by millisecond. And so, for example, we can imagine that there's a stimulus at this 0 milliseconds. So let's just say that's table. And from this device, we can then record the strength of the magnetic field millisecond by millisecond. And it turns out that the electrophysiological response to a word like this has a kind of a characteristic shape. So most individuals will show a certain component structure to the response that establishes a decent enough a generalization that we can kind of study the functional roles of those different stages of the response. And so specifically, so here I'm showing the characteristic magnetic fields associated with these peaks that we see in the sensor recordings. And given that there's enough of a generalization about the shape of this response, these components have labels that are used in the literature when we talk about them. So here, the first peak is something that we usually refer to as the visual M100. So remember, in the auditory domain, we had an auditory M100. So this is a similar early response, but now just elicited by visual stimuli, and it localizes in the visual cortex. Here, I'm just showing you the magnetic field distribution. Red blob is outgoing flux. Blue blob is flux re-entering the brain. Then we have the next peak, it's called M170. And so here again, that just refers to the fact that it peaks roughly at 100 milliseconds. 170 refers to the fact that it peaks roughly at 170 milliseconds. And then a couple of later components as well that are pretty consistent across individuals. So now we have nice temporal resolution, some understanding of the general shape of the response. We can ask the question, well, when exactly in the stream of responses sensitivity to letter strings emerges? And we have a pretty consistent finding from the literature that tells us that it is most robustly observed at this M170 stage. So the M170 response is our clearest MEG correlate to the visual word form area. And in the EEG literature, you can also find an N170 literature. So it's a negativity that is observed roughly around 170 milliseconds. So what do we know about this response then more specifically? A lot of what we know about it comes from the research of this group. Here's an original research, a paper from Tarkiainen and colleagues, 1999, from the journal Brain. And so here's the typical type of experimental design that they would use in order to study letter string sensitivity in the MEG response. So here, as you can see, one manipulation pertains to whether the person is seeing a string of letters or not. And so this actually happens to be the Finnish word for money, raha, but it's a letter string. And we're comparing it to a visually similar string of symbols. So these are, have some of the same low-level characteristics as the letters, but they're just symbols. And then in addition to this stimulus content manipulation, we're varying a couple of things that pertain to the sort of the physical properties of the stimulus. So first of all, they're varying the size of the stimulus. Okay, so that's something we're not really interested in, but it's something that certainly should modulate low-level visual responses. And then we're also varying the noise level of the stimulus. So the letter string stimuli are also embedded in different degrees of visual noise. And so in these cases, it's harder to make out what the letter string is. So we're sort of manipulating the effortfulness of the letter string perception. So what have we learned from these kinds of designs? Well, we've learned that at the M170, activity is stronger for letter strings than for symbol strings. So that is now suggestive of a correspondence to the visual word form area. We also have learned that activity at the M170 is delayed or decreased for less detectable letter string stimuli. And so that, again, suggests that this is reflecting the kind of work that you put into to actually observing the letters. And finally, the activity does also localize in or near the visual word form area, as suggested by the hemodynamic literature. So as we've learned from MEG data, from these magnetic field distributions, we can actually pretty accurately localize the underlying current, so exactly what neural activity is generating that field. And so, for example, in this study, you see a pretty consistent cluster of localizations in an area that's consistent with the visual word form area, as we've learned from fMRI. Okay, so now we've looked at 3 different methods, and they're kind of all pointing in the same conclusion in a pretty nice way. So in fMRI, we see visual word effects in the left fusiform gyrus. In MEG, we see similar effects in the left hemisphere M170 response, which localizes consistently with the visual word form area. And also aphasia evidence is consistent with this. So if you have damage in the left fusiform gyrus, it does impair your reading typically, and the characteristic disorder is called pure alexia, so and exhibits as letter by letter reading. I have a little parenthetical here about something that points to more contemporary results. So in fact, we now know that you can get visual word form effects even earlier already at the M170 stage, but we don't have to worry about that here. Okay. So I told you, I told you about the FFA, the fusiform face area, and then I was just like, well, it doesn't seem really plausible that We could also have a visual word form area because we just don't have enough evolutionary time to have evolved such an area, but now it does seem like we have such an area. So how can we understand this? How could our brains get this way? Well, it seems to be the case that the human visual system in general has kind of a readiness to specialize particular regions for the perception of specific types of categories. But exactly why we all seem to represent visual word forms exactly in a similar region is a puzzle in the context of that. There is an interesting, relatively recent proposal that offers one way to think about it. And the way is that the visual word form area is in some sense piggybacking on a region that is actually a more general object recognition area. So specifically, we seem to have an area in our brains that is pretty much in the neighborhood of the visual word form area, where we represent line junctions, and specifically line junctions of the sort that most orthographic systems employ. And so it seems to be the case that instead of having a really dedicated visual word form area that just sort of comes out of the blue, our brains all kind of co-localize visual word form perception next to this area that's already representing line junctions. And so here we're basically explaining the systematic localization in terms of this older ability, an older ability for which we sort of have enough evolutionary time that we might have actually developed a patch of cortex that's dedicated to it. And this is a form of a hypothesis that's more generally called a neuronal recycling hypothesis. So you take some cortex that does a kind of a more older basic ability. By older, I just now mean evolutionarily older, and then you kind of tweak it a little bit so that it can do some newer tasks. There's some very interesting recent evidence from monkeys that lends further plausibility to this hypothesis. So here they're actually kind of having the monkeys read. So they are presented with letter string stimuli, and these recordings from the monkey cortex actually lend some further evidence for the idea that we might have this precursor of orthographic processing in the inferior temporal cortex. So perhaps that is how our brains got this way. And next we'll talk about dyslexia. So when something goes wrong in the reading system.